Hello student, in this video we will discuss class 12 biology chapter number 2, that is reproduction in flowering plant. And before we start, let us see what we are going to study in this chapter. So in this chapter we will discuss about pre-fertilization, fertilization, post-fertilization event in plant. And when you come to pre-fertilization, here we will study about the structure of male and female reproductive part, about the formation of male and female gamete, that is gametogenesis. So here we will study microsporogenesis and megasporogenesis. This will followed by the gamete transfer. Here we will study pollination. And this pre-fertilization event will be followed by fertilization. And in case of plant, double fertilization takes place. So in this chapter, we will discuss what is double fertilization and how double fertilization takes place in plant. And this fertilization will be followed by post-fertilization event. So here we will discuss about the development of endosperm, embryo, fruit and seed. So these are the things that we are going to study in this chapter. And when we come to plant, all this event will take place in a flower. So before we start the class again, we have to remind something about the flower, mainly about its morphological structure. So flower, actually it is a modified shoot and it is usually modified to perform sexual reproduction. That is, flower are the site of sexual reproduction. And morphologically, flower consists of 4 main parts. And here the outermost green part is known as calyx, which is represented by sepal. Next, colorful part of the flower, it is corolla and is represented by petals. And next part is endoecium, represented by stamen, and we know that stamen consists of filament and anther, and this anther will produce pollen gland, which will produce male gamete. And the innermost part of the flower is known as gynoecium, which will be represented by carpel or pistil, and it consists of stigma, style and ovary. And inside the ovary, what is present? Ovule is present, and we know that in that ovule, female gamete egg is present. So in a flower, this androecium and gynoecium will represent the male and female reproductive part of a flower. So in this chapter, we will discuss in detail about androecium and gynoecium. Okay, so this is about flower. So now coming to our topic, here in this video class, we will study about the structure of male reproductive part and microsporogenesis. Male reproductive part. We know that androecium is the male reproductive part of a flower, which is represented by stamen. And we also know that stamen is composed of 2 parts, that is anther and filament. Now coming to anther. In most of the flowering plant, this anther is a bilobe structure, okay? And each lobe contains 2 chamber each. And because of this, anther are known as bithecous or dithecous. I will repeat. Anther, okay, in most of the flowering plant, this anther is a bilobe structure, and each lobe contains 2 chamber each. Because of that, anther are known as bithecous or dithecous. Next, in this chamber, what happened? Microspores are produced. Because of that, the chamber can also be known as microsporangia. And here, if I say zea, it means plural form, zea means singular. And that microspore will later develop into pollen gland. That means in this chamber also pollen gland are produced. That's why the chamber can also be known as pollen sac. That means the chamber which is present in the anther can be either called as microsporangia or pollen sac. Okay. Next, when you cut a section of a young anther before pollen grain are produced, you will find that the chamber, that is microsporangium, is surrounded by 4 wall layer. And these 4 wall layer are epidermis, endothecium, middle layer, tapetum. And we also know that epidermis, it is the outermost cell layer of the plant part. And next to epidermis, you will find a single layer of cell known as endothecium. And inner to endothecium, you will find 2 to 3 layer of parenchymatous cell known as middle layer. And this tapetum represent the innermost wall layer. And some important features of tapetum are the cells of tapetum are usually multinucleated. That means more than 1 nucleus is present, and the nucleus are polyploidy. That means the nucleus have more than 2 sets of chromosomes. Here, if I say one set of chromosome, it means haploid. If I say two set of chromosome, it means diploid. But here, the nucleus have more than two set of chromosome. That means the nucleus is polyploidy. Okay. Now coming to the function of tapetum. Here, the tapetum, they will provide nourishment or nutrition to the developing pollen grain. Actually, in this chamber, pollen grain will be produced. Okay. So in the developing pollen grain, nourishment will be provided by this tapetum. So all these four wall layer have their own function. Okay. So here epidermis, outer three epidermis, endothecium, middle layer, they perform the function of protection. On the other side, this tapetum, innermost wall layer, it provide nourishment to the developing pollen grain. Next, this chamber, okay, in the young anther, this chamber, that is microsporangium, will be filled with a homogeneous, completely arranged cell known as sporogenous cell. Actually, what happened here, this sporogenous cell will divide to form microspore, and then it develop into pollen grain, and this pollen grain will produce male gamete. Here we will discuss how this sporogenous cell develop into microspore, and then pollen grain, and then into male gametes one by one. Microsporogenesis. So during anther development, what happened? The sporogenous cell, which is present inside the chamber of the young anther, they divide repeatedly and form microspore mother cell. And this microspore mother cell will undergo meiosis to give rise to what? Four haploid microspore. So the process of formation of this microspore from microspore mother cell through meiosis is known as microsporogenesis. Okay, I will explain the point again. Actually, what happened here? The sporogenous cell, which is present in the chamber of the anther, they will function as microspore mother cell, which undergoes reduction division, that is meiosis, to form microspore. And we know that during meiosis, chromosome number will be reduced. So microspore will have haploid number of chromosome, plus during meiosis we know that four daughter cells are produced. So you will get four microspore. So you will get what? Four haploid microspore. So the process of formation of haploid microspore from microspore mother cell through meiosis is known as microsporogenesis, and this process takes place in anther. Okay, and these four microspore remain clustered together, forming a tetrahedral tetrad. But when the anther mature, these four microspore will separate from each other, and each microspore will develop into pollen grain. So this is how pollen grain are developed from microspore. Now coming to pollen grain. When you come to pollen grain, the most important point is that pollen grain are always haploid. Why? Because pollen grain develop from haploid microspore. And this pollen grain also represent the male gametophyte of a flower. And pollen grain is also surrounded by cell wall, which consists of two layers, outer exine and inner intine. And this outer, that is exine, it is thick and it is made up of sporopollenin. And actually sporopollenin is an organic substance which is highly resistant to chemical and biological decomposition. And sporopollenin can withstand high temperature, it can withstand strong acid, and even it can withstand strong alkali also. And till now, no enzyme has been discovered which can degrade sporopollenin. So because of presence of sporopollenin in the pollen grain, so definitely pollen grain will be highly resistant to chemical as well as biological decomposition. Pollen grain will be able to withstand high temperature, strong acid as well as strong alkali, and pollen grain will not be able to digest by any kind of enzyme. And as pollen grain cannot be digested by any enzyme, the inner component of the pollen grain can be well preserved as what? As fossil. That means pollen grain can also be preserved as fossil because of presence of sporopollenin. So these are the importance of presence of sporopollenin in pollen grain. And at certain place, what happen, this exine may be totally absent or it may be very thin, and that particular region will be known as germ pore. And in this germ pore, pollen tube will be produced. Last one, intine. We know that intine, it is the inner wall, it is thin and it is composed of cellulose and pectin. And when pollen tube is produced, the pollen tube will be surrounded by intine. That means the wall of the pollen tube will be made up of intine. So this is something about pollen grain. Okay, so before we wind up, let me remind you of what we have learned in this video class. So in this video class, we have learned about the structure of anther, about microsporogenesis, and about some features of pollen grain. And in the next video, we will discuss how this pollen grain produce male gametes. So don't forget to follow up the next video. Okay, and thank you very much for your attention.